Welcome to the next session on tricyclic or Krebs cycle or citric acid cycle in the carbohydrate metabolism series. This session will now continue on looking at what happens after glycolysis to the pyruvate as it enters the mitochondria as acetyl-CoA. So the citric acid cycle is also known as the tricarboxylic acid cycle, or TCA, or the Krebs cycle. This is the pathway that most cells use to create energy, ATP, and it is occurring in the mitochondria, whereas glycolysis is occurring in the cytoplasm. So further on, we'll talk about how that pyruvate gets into the mitochondria for the Krebs cycle. But this is the final common pathway for oxidizing carbohydrates, lipids, and proteins. So all roads lead to this cycle, whether it be carbohydrates, whether it be protein coming from muscle or other areas, or fatty acids. So all of those are metabolized to acetyl-CoA or intermediates of the cycle in between. So the first step is getting the pyruvate from the cytoplasm at the end product of glycolysis into the mitochondria, which is the acetyl-CoA. And this is an enzyme known as pyruvate dehydrogenase that catalyzes all of this. It requires multiple cofactors and some coenzymes to make it happen. So the first step in E1 is where the pyruvate is released as CO2, and this attaches to thiamine pyrophosphate, one of the cofactors that's required. As the name implies, thiamine is required for this. So one of the areas that we'll see this problem is in thiamine deficiency, where people are not able to do this process very well, and hence are fatigue and not enough energy. So the next phase that happens is where the acetyl-CoA goes into the fully reduced form. And then the final step in this process is the oxidized form of this whole complex, which then becomes acetyl-CoA. The acetyl-CoA releases carbon dioxide in this process, and this now can enter the mitochondria to enter the Krebs cycle, or the TCA. So this is an overview of the TCA. It requires multiple coenzymes along the way, usually NAD+ and FAD, that are reoxidized through this electron transport chain. This will be covered in a separate session to form ATP there. Gluconeogenesis, lipogenesis, and also converting amino acids, they're all connected to this cycle. So let's now look at some of these steps. So the first main area is this acetyl-CoA coming in to form combining with oxaloacetate to form citrate, hence the name citric acid cycle. The next few reactions are going to metabolize that citrate along the way to generate ATP, and then also to come back again and regenerate. So the cycle starts with the reaction of this acetyl-CoA forming the citrate. In the next few reactions, as that CO2 is released, oxaloacetate is regenerated, as I mentioned previously. And then the final step is malate, which regenerates that oxaloacetate. So let's focus on one of these enzymes, which is succinate dehydrogenase, which is a very unique enzyme. It's actually on the inner membrane of the mitochondria, and it catalyzes that first dehydrogenase reaction to form fumarate. It is an electron carrier and is also part of the complex that converts succinate to fumarate in addition to participating in the electron transport chain. This will be covered in a future session, but this is the basic complex of the electron transport chain, also known as oxidative phosphorylation or the electron transport system. It goes by multiple names. This is on the inner part of the mitochondria, the inner membrane, and it is generating ATP in the end. It requires oxygen to do this. On the left and the right, these steps are shown there. It'll be covered in detail a little bit more. But in general, what we're focusing on in this session is that succinate dehydrogenase is part of the complex 2 of the electron transport chain. So there is some overlap here between the Krebs cycle and the electron transport chain. So back to the TCA, step 1 is forming the citric acid. Step 2 is isomerizing that citric acid to cis-aconitate. And then the next enzyme in the next step 3 is forming isocitrate. During this step, NAD is then reduced to form NADH. This is also a regulatory enzyme here in the Krebs cycle with forming NADH, because NADH can go back and inhibit if there's too much overproduction and slow down the Krebs cycle here. The next step is the formation of alpha-ketoglutarate. and it releases a molecule of carbon dioxide in the process here. So the remaining 4-carbon molecule here is the succinyl-CoA, which is very unstable. Alpha-ketoglutarate dehydrogenase is required for regulating also the Krebs cycle. So in step 5, the succinyl-CoA, the CoA of succinyl-CoA is replaced by phosphate and just forms succinate. In this step here, it's either going to form ATP or GTP, depending on which cell is doing this process. And the end result here is succinate. The final steps here are regenerating oxaloacetate, so we can continue this cycle over and over again to form more ATP. So in step 6, the formation of fumarate, there's another FADH2 formed here. And then in step 7, reforming oxaloacetate, another NADH is formed there. So this concludes the cycle of TCA. Please subscribe to ACM, the YouTube channel, for further updates, as these are uploaded there. Thank you.